Thank you again, Brooker, for providing me the wonderful opportunity to speak with everyone at the symposia today. Hi, everyone. My name is David Roberts. I'm a senior graduate student at the University of Wisconsin-Madison. And today I'm going to be talking to you about the structural O-glycoform analysis of SARS-CoV-2 enabled by top-down mass spectrometry and trapped ion mobility. So to begin with, I want to first introduce a little bit of the type of research conducted in the G group. So broadly speaking, our work is highly interdisciplinary in nature and cuts across the traditional boundaries of chemistry, biology, and medicine. Our group aims to understand the molecular and cellular mechanisms underlying cardiovascular diseases via systems biology approaches, all featuring cutting-edge, high-resolution, top-down mass spectrometry. And one of the major focuses in our lab is the top-down proteomics. And so to motivate the topic, here I'm showing a revised illustration of the classical central dogma of biology, DNA going to mRNA going to proteoforms, a term which encapsulates all proteins and protein families that can be a result of alternative splicing, mutations, and post-translational modifications arising from a single gene. So with the success of the Human Genome Project, which occurred 20 years ago, and as can be seen with this exponential increase in complexity as we move from each stage of information flow, researchers have come to recognize that much of the complexity afforded by our biological machinery is at the level of protein variation rather than the gene. And so our lab uses top-down mass spectrometry, which is capable of revealing the molecular variations of proteins at this proteoform level. And specifically, it turns out that post-translational modifications are pretty crucial for understanding the dynamic regulation and interactions between the S protein and ACE2. So I'll start with first showing a little bit of some motivation for that. So you might recall that the 2002 SARS actually also occurred, and the 2019 current novel coronavirus, in comparison, actually don't differ too much just by a primary sequence metric. In fact, a structural analysis of these two things reveals that there's a lot of similarities in homology. However, there's a lot of complexity that's hidden underneath this, guys, because the same therapeutics that we had developed before in 2002 to target SARS, the original SARS, no longer seem to work very well for the 2019 coronavirus. And it's raising a very important question as to why is it that exactly this is happening. And so recent work by Max Crispin's group, as shown here, has shown that that there are actually a lot of detailed post-translational modifications that can occur on both the S protein as well as ACE2. But here, specifically showing the S protein, you see these colored heat maps. These are various sugars or glycan structures, specifically N-Glycans, that are being decorated along the protein surface. And even just here, you can see that it's quite complex. There's a lot of different modifications, and this is only showing the oligomannose mapping. Taking a step further, Romy Amaro's group at UC San Diego have recently shown that post-translational modifications, in fact, glycans themselves, likely induce viral receptor interactions. And as you can see just from this little zoom out of the S protein ACE2, those blue spheres you're seeing around the protein surface is actually just representing the glycans. And so it's really a jungle of many different modifications, and it really raises the question of how these PTMs influence the actual viral biology. And this is not necessarily a new concept. Classically, viruses are heavily regulated, and their functional roles are heavily dictated by their glycosylation, ranging from attachment and entry, adaptive immunity, spread and innate immunity, and assembly and exit. And so we come to understand that glycosylation on viral proteins tends to play critical roles in how they perform their function when introduced to the body. And more specifically relevant to SARS, recently work by Lance Wood's group have shown that SARS-CoV-2 and ACE2 glycans are critically involved in true receptor interactions, but not just at the protein surface level, but potentially the glycosylation themselves could be mediating this binding. And so altogether, this underscores the level of molecular characterization and the sort of role that glycan structures may have for understanding not only how SARS interacts with human receptor ACE2, but potentially changes or variations in these glycan structures that we might be seeing from emergent SARS-CoV-2 variants may give new reasons to try to reconcile these glycan and post-translational modification differences. And so, from our point of view, we understand that this is a huge challenge, but also from a scientific point of view, this presents new and exciting opportunities to potentially understand some of these molecular details. And so specifically, we understand that post-translational modifications, specifically glycans, are heavily implicated in this S-ACE2 structure-function interaction. And so we understand it's really crucial to understand these things at a molecular level and to help better design rationally designed therapeutics. More specifically, traditional structural methods that can be used to illuminate this problem, such as X-ray crystallography and cryo-EM, are very helpful for trying to understand some of the protein interactions, but they're not completely amenable for comprehensively characterizing these glycoproteins. And this is due to the intrinsic flexibility and heterogeneity of these sugars. They're just wobbling all the time, and there's no real good way to get one crystal that reflects the entire stoichiometry of what's going on. And so this is where me and the rest of our group believe that top-down mass spectrometry can hold the key to trying to provide some of this molecular elucidation at the proteoform level. Specifically, we believe that top-down MS can provide the key molecular insights to reveal the complex structural landscape of heavily modified proteins, namely the S protein and potentially ACE2. And we feel this way because what a lot of the things that we've demonstrated in the GUG group, specifically using top-down, is the idea that we can use it to provide a bird's eye view of complex proteins and their various modifications. So recently, we've developed a top-down proteomics platform that is capable of providing high-resolution proteoform-resolved molecular fingerprints of a cardiac biomarker, so cardiac troponin I, a gold standard biomarker used in the clinic. And we've shown that top-down MS is capable of revealing all of these diverse cardiac troponin I proteoforms in a single spectra. And when used to compare between different stages of disease pathology, can potentially be useful to establish proteoform pathophysiology relationships. So we have some evidence already that top-down is capable of wrangling some of this intrinsic complexity that occurs at the protein level. And more specifically, when applied to SARS-CoV-2, which we understand is a viral RNA protein, so it's essentially just a protein bundle with an RNA payload, the S protein, which is the surface receptor of the SARS-CoV-2 virion, specifically engages with ACE2, but even more so, the exact domain, the S-RBD regional binding domain of the S protein is what specifically interacts with our host receptor. And so conventional methods to understand this glycosylation, as I've mentioned, rely on using glycopeptides or other means to engage in these molecular structures. But here we can use top-down to gain this holistic and intact view of all of these changes. And specifically, what I'll show here and the focus of today's talk is on utilizing a hybrid native TIMS approach with an ultra-high-resolution Fourier transform ion cyclotron approach to not only understand the structural heterogeneity of the S-RBD, but also simultaneously characterize its molecular structures. And that workflow is better illustrated here, where we're calling this a hybrid top-down approach, where on one hand, we take the S-RBD and we utilize timsTOF to provide native ion mobility analysis of the S protein, specifically using trapped ion mobility. This allows us to both get an idea of the glycoform structural landscape, as well as some of the structural heterogeneity associated with each of these individual glycostructures. And on top of that, we leverage the ultra-high-resolution ability of the Fourier transform ion cyclotron Solarix 12 Tesla Bruker mass spectrometer to provide an isotopically resolved and very high accuracy understanding of each of the different glycostructures with the ability to characterize its sequence and sites. And so we begin by first looking into exactly how we prepare the sample for this type of removal. And in this work, we focus mostly on the O-glycoform side. There's been a lot of good work demonstrated already by others on understanding N-glycosylation, but O-glycosylation, the other side of the glycomics world, is relatively understudied. And so here we're using this approach to provide new information that could potentially illuminate some of these lesser studied structures, but still potentially virally important. And so here is a visualization of the before and after treatment with Paynease F to remove N-Glycans. This constitutes about a 10 kilodalton loss in molecular weight. And really nicely, after removing all of these N-Glycans, not only were we able to resolve the O-glycan forms in their intact state, but we were able to do this natively and achieve isotopic resolution. It's actually quite a important point here to stress how complicated even just these regional binding domain proteins are on the SARS-CoV-2 spike, because even without removing the N-glycans, you actually see in the top left that there's a huge heterogeneity spread of various glycoforms that makes analysis rather challenging. And so after removing these N-glycans and being able to resolve this natively, we move over into the native TIMS MS analysis to try to wrangle in some of this glycoform heterogeneity. And so specifically, what we used is native TIMS, which was sensitive and able to both separate these S-RBD glycoform species by ion mobility, but also help to resolve some of the conformational heterogeneity. And so what you're seeing here are regional separations in ion mobility space on that heat map, which actually correspond to a native monomer and dimeric S-RBD species that we were able to resolve. And so using ion mobility, we were able to characterize the collision cross-sections of the various charge states associated with this species and try to understand a little bit more about some of this conformational landscape in the gas phase. In doing so, we were able to not only isolate specific regions of the S-RBD in its native state, but also take advantage of the fact that we can further characterize those different species by MSMS. And so here what you're seeing are examples of using the timsTOF to specifically isolate in mobility space some region of interest corresponding to, say, one of the charge states or one of the monomeric or dimeric species of the S-RBD, and then fragment to try to understand where the sites of these modifications are and what exactly are the forms. And so at the left, you're seeing an example of some native fragment ions that we use to confidently assign the O-glycosite of this S-RBD specifically to the threonine 323 residue. We found that that was the only occupied site of all of these O-glycan structures. And more so, we were able to even look into the native ion mobility separation of the various O-glycan structures, as you can see on the right, and map this relative to the crystal structures that have been shown for the non-glycosylated S-RBD, and show that these O-glycosylations tend to provide enough of a structural difference that their ion mobility and collision cross-section is notably enhanced. So going a step further, now that we were able to look into some of the native conformational landscape of this protein, we moved into analyzing the proteoform-specific O-glycan structures. And to do this, we use the ultra-high-resolution Fourier transform ion cyclotron resonance MS. So to begin, we're showing here first a kind of full survey view of the S-RBD in its entirety after the Paynease F treatment. If you look into each of the individual forms, and in this particular case, I'm showing the most abundant charge state, you can see that each and every one of the individual glycostructures are in fact baseline resolved. And this is great because now we can take each and every one of these proteoforms and perform an isolation of them specifically using narrowly defined quadrupole widths. And with each and every one of them, we can begin to fragment them progressively, going from softer to harsher ionization, so that we can preserve as much of the intact structure as we can, while making sure that we still are able to resolve their sites and the exact locations of each of the glycans. So what you're seeing here is sort of a picturesque view of what goes on for each and every single one of these O-glycoforms. There's a site-specific isolation, there's a specific quadrupole isolation, and we progressively ramp up the collision energy such that we can basically put the pieces of the puzzle back together, connect the dots, and really begin to understand what both the intact glycan form was under a soft ionization and what the and prove the actual combinatorial makeup of these various O-glycan structures. And so this represents a very powerful, high accuracy characterization of these highly heterogeneous glycoproteins, which even now is still very challenging for a lot of different applications. So altogether, we found that using this hybrid top-down approach, we were able to, in fact, completely structurally elucidate the S-RBD O-glycoform heterogeneity and provide this sort of bird's eye view for each and every one of the O-glycoforms, with the simultaneous ability to characterize the molecular abundances. So not only do we understand now the glycan structures and we understand the site, specifically threonine 323 in our case, but we are also able to look at the molecular abundances, so the molecular stoichiometry of these various glycan forms. And using this information, we were able to reveal that these core 1 O-glycan GalNAc neuremic acid structures represent actually about 65% of the total O-glycoform abundance. And so maybe going a step further, future studies can take advantage of some of these site-specific or glycoform-specific information to perhaps better understand or even target specific glycan features for targeted therapeutics. And so what I'm showing here is sort of this kind of landscape now, a structural landscape of all the O-glycosylation that has been revealed by top-down MS for this SARS-CoV-2 protein. And so I hope you see that this can be a potentially very powerful approach for characterizing emergent S-RBD variants towards future structure function studies. And so with that, I would like to thank everyone for listening, and especially I would like to give some thanks to my dual advisors, Professor Songjin and Ying, for providing me the opportunity to speak with you today. I'd also like to especially thank our collaborators, Dr. Alan Brazier and the rest of the Brooker team, for their continuous support. Being a member of 2 different groups feels like you're a member of a very large family, so I'm eternally grateful for that always. Special thanks to my funding sources, the AHA, the NIH, and thank you all so much for listening. Please feel free to reach out to our group anytime, and I'm happy to answer any questions at the end of the session.